Hello guys, I am Shubham from Short Tutorials. In this video, we will be seeing prokaryotic transcription PYQs. So before we start, subscribe to our channel and press the bell icon to get notified whenever new videos out. This is absolutely free from your side, but it will help us grow. So all about prokaryotic transcription we have discussed. You will find the playlist as molecular biology. Okay? All the topics we have discussed. First question. Which one of the following statement is not correct? Okay, not correct हमें आइडेंटिफाई करना है. टुगेदर विथ प्रोटीन्स RNA provides a site for polypeptide synthesis. So this is the correct statement, right? Polypeptide synthesis के लिए site provide करता है RNA together with proteins. Second point, all DNA molecules are unbranched polymers of nucleotides. So this is also correct. Unbranched polymers means linear molecule होता है DNA. Only it is wounded around itself. So this is also correct. DNA is synthesized in a 5' to 3' direction, while RNA synthesis occurs in a 3' to 5' direction. This is incorrect statement. Okay. Very important. You have to know DNA synthesis 5' direction में होता है. तो मतलब replication. While RNA synthesis occurs in 3' to 5' direction. So this is also same, right? This is also same. So ये third option, this will be our answer. A tRNA anticodon may pair with more than one codon. This is also correct. Okay, incorrect हमें identify करना है. So replication as well as transcription, translation all occur in this direction only. In bacteria, heat shock response is primarily controlled by. So sigma factors are of different types. Okay, sigma factors अलग अलग प्रकार के हैं. And जो heat shock के लिए रिस्पॉन्सिबल है, that is sigma 32, option 2. Okay, heat shock response controlled by sigma 32, right? So there is a list of the sigma factors. Sigma 70 main sigma factor है, that is for proper promoter binding. Okay, specificity के साथ bind होता है. Then sigma S is there for stationary phase some stress response. Okay, S stress response. 32 heat shock. Then E is periplasmic extracellular protein. Sigma 54 hydrogen assimilation. Sigma F is flagellar synthesis chemotaxis. And sigma FED is ion metabolism transport. Okay, so this is a very important slide. You can make a screenshot, right? I have not shown this in the transcription video. So note this. And यहाँ से you can see there are बहुत क्वेश्चंस आए हैं इस एरिया से. Repetitions, you can see lot of repetition. A mutation in which one of the following sigma factors may be a possible cause for E. coli failing to adapt in response to thermal stress. Okay, thermal stress, right? Thermal stress is again heat shock only. Thermal is related to heat, right? So same type of question है sigma 32. क्वेश्चन का मीनिंग क्या है? ऐसा सिग्मा फैक्टर जिसमें म्यूटेशन है, म्यूटेशन इन अ सिग्मा फैक्टर दैट कॉज, उसके कारण से एडाप्टेशन नहीं हो सकता इन द रिस्पॉन्स टू थर्मल स्ट्रेस. इट कैन नॉट adapt itself. So that sigma factor which is responsible for this is sigma 32. Presence of sigma factor 70 in Escherichia coli facilitates, okay, what? Sequence specific localization of RNA polymerase on the promoter region of genes. Absolutely correct है option 1. ये sequence specific binding होती है, right, with the help of RNA polymerase on the promoter region, right. Next, statement phosphorylation of C-terminal tail of RNA polymerase, no. Interaction of beta subunit of RNA polymerase with alpha subunit, no. Interaction of omega subunit with beta, beta dash alpha 2 complex, no. So this is also a slide about different RNA polymerase factors. RPOA is 2 alpha subunits, enzyme assembly, promoter recognition, bind some activators. Then beta, beta subunit, beta dash catalytic center है. Then omega subunit is promoter specificity. Okay, sigma factor. Omega subunit helps sigma subunit to bind. Right. Sigma factor, sigma is promoter specificity, and omega is helping for its binding. Now omega help करता है sigma को to bind the promoter. Which one of the following ensures stable binding of RNA polymerase at the promoter site? Again, same question. Stable binding of RNA polymerase. Stable binding किस एंजाइम से होगी? तो DNA photolyze, no. This is involved in repair. Sigma factor, yes. DNA glycosylase again repair enzyme. REC भी repair. Homologous recombination में. So answer is option 2. So you can see so much repetition is there. One of the mechanisms used by bacteria for adaptation to change environment. Okay, adaptation change environment. जो environment change हुआ है उससे adapt होना है. Is altering transcription of their genes. Transcription को alter करना. In this regard, which one of the following is not found in bacteria? Bacteria में नहीं होता. Option 1, a gene with 2 different promoters for expression in different conditions. मतलब जीन है तो 2 different promoters हो सकते हैं for expression in different conditions. Why not possible? So this is correct. Okay, we have to found not found. जो incorrect statement है तो ये हो सकता है. Okay, हो सकता है. Basically this is about gene regulation, we can say. Okay, gene regulation in bacteria. मतलब ट्रांसक्रिप्शन जीन का कंट्रोल कैसे कर सकते हैं. ओके, तो यू कैन डू दिस 2 डिफरेंट प्रमोटर्स से हो सकता है फॉर डिफरेंट कंडीशन. सेकंड ऑप्शन, यूज ऑफ डिफरेंट सिग्मा फैक्टर्स फॉर ट्रांसक्रिप्शन ऑफ जीन्स. यस, अभी हमने क्वेश्चन इतने देखे, टेबल देखा, कितने डिफरेंट टाइप्स ऑफ सिग्मा फैक्टर्स है. तो different sigma factors are used for controlling transcription. Possible. Expression of alternate alpha, beta and beta dash subunit. Okay, alpha, beta and beta dash subunit का alternate expression possible नहीं है. Why? Because this consists of core enzyme. Okay, core enzyme sigma के साथ जब जुड़ हो जाता है that is holo enzyme. So core enzyme alternate expression is incorrect statement. Next statement, expression of anti-sigma factors. Yes, possible. So 1, 3, 4 ये जीन रेगुलेशन में पॉसिबल है इन द बैक्टीरिया, बट जो थर्ड ऑप्शन है दैट इज नॉट पॉसिबल. सो दिस इज नॉट फाउंड इन बैक्टीरिया. ओके, बिकॉज अल्फा, बीटा, बीटा डाई सब यूनिट इज अ कोर एंजाइम का पार्ट है. दे आर पार्ट ऑफ कोर एंजाइम. So their alternate expression is not possible. So they have to express, because we know alpha is for promoter recognition, beta, beta dash catalytic है. Okay, so these are important and they should express at same time. Anti sigma factor क्या हो सकता है? This can control the gene, right? अगर sigma 70 efficient binding, okay, it does efficient binding. Okay, means what? Gene transcription increase होगा, right? Enhance होगा. But if there is a anti-sigma factor, this will block this. Okay, मतलब ऐसी condition है said in the question, environment changes suddenly. So this anti-sigma factor can block this sigma. Okay, sigma को block कर सकता है, and then there will be no gene regulation or transcription पे नहीं होगा when the anti-sigma factor is expressed. So this is possible, right? Which one of the following statement is not a correct feature of Escherichia coli RNA polymerase? So again, not a correct feature. First option, presence of sigma subunit along with alpha 2 beta beta dash omega core RNA polymerase is required for promoter specific binding. करेक्ट है, करेक्ट है. We have to see the not correct, right? Presence of the sigma subunit along with same core RNA polymerase is not necessary for the core RNA polymerase to bind to the DNA template. Is not necessary. You can get confused by this statement. So I will explain. This is also correct. ये भी करेक्ट स्टेटमेंट है. Why? सिग्मा फैक्टर नेसेसरी नहीं है. Okay, this is not necessary. As we saw, alpha alone can bind. Okay, as we saw, enzyme assembly, promoter recognition. Okay, promoter को recognize कर सकता है and it can bind some activators. सिग्मा फैक्टर क्या करेगा? It binds with specificity, but only binding इससे भी possible है, right? Alpha subunit can do the binding, so it is not necessary. So this is the correct statement. Mutation in beta subunit in its rifampicin resistance defining region confer rifampicin resistance phenotype. This is also correct. You have to remember this. Beta subunit में अगर mutation है, okay, if there is a mutation in beta subunit, then results in the rifampicin resistance phenotype. Why? Because beta subunit has a rifampicin resistance defining region. Okay, beta subunit में present है. Okay, rifampicin resistance defining region. So mutations in alpha subunit in this rifampicin resistant defining region confer rifampicin resistant phenotype. Wrong होगा. And this will be हमारा आंसर because we have to see the not correct feature. Okay, correct feature नहीं है. 3 and 4 में से एक ही करेक्ट हो सकता है, राइट? सो बीटा सब यूनिट में म्यूटेशन अगर है तो रिफाम्पिसिन रेजिस्टेंस होगा, नॉट इन अल्फा सब यूनिट. तो हमारा आंसर फोर्थ ऑप्शन है. एक्टिनोमाइसिन डी inhibits the process of transcription in both prokaryotic and eukaryotic organisms. Right? Actinomycin D antibiotic transcription inhibit करता है in the both prokaryotic and eukaryotic organism. The following statements are made about actinomycin D mediated inhibition of transcription. तो कैसे करते हैं inhibition? That we have to see. Which of the combinations of the above statement is a true representation of mechanism? Okay, mechanism हमें identify करना है of actinomycin D mediated inhibition. A option, actinomycin D inhibits transcription from double-stranded DNA template by either E. coli or yeast RNA polymer. This is 100% correct statement. A करेक्ट है. हमें ट्रू ही आइडेंटिफाई करना है. डोंट गेट कंफ्यूज, क्वेश्चन को अच्छे से रीड करना है, राइट? समटाइम्स यू मिस द नॉट और इनकरेक्ट स्टेटमेंट, राइट? मिस हो जाता है. केयरफुली रीड करना है. बी स्टेटमेंट इज एक्टिनोमाइसिन डी inhibits transcription from a single stranded RNA template by eukaryotic viral RNA polymer. Oh, correct है. Actinomycin D inhibits transcription from single stranded. Oh, ये भी wrong है. Okay, this is also immediately after viral entry. Wrong है ये भी. Actinomycin D inhibits transcription from double stranded RNA template by eukaryotic RNA polymerase 2. This is also incorrect. Correct statement है only A. The correct option 1 correct है. A, ABD wrong, ABC. C statement गलत है. As well as B and C. All of the following statements about bacterial transcription termination are true except, again we have to identify incorrect statement. Okay, क्या लिखा है? All statements are true except, right? Except सब एक को छोड़ के बाकी सारे करेक्ट हैं. So some terminator sequences require raw protein for termination. करेक्ट है ये करेक्ट हमें नहीं देखना. Inverted repeat and T-reach non-template strand define intrinsic terminator. करेक्ट है ये भी. इन्वर्टेड रिपीट चाहिए एंड T-reach non-template strand मतलब कोडिंग स्ट्रैंड में T-reach रीजन चाहिए. ओके, सो दैट जो mRNA होगा उसमें यू 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 रीच, राइट? यू रीच. So these are correct. Row dependent terminator may possess inverted repeat elements. ये भी करेक्ट है. If this क्या करेक्ट ये रॉन्ग होगा तो ये हमारा आंसर होगा. Okay, NUS A is necessary for intrinsic transcription termination. So we have not seen about NUS A. हमने ये 3 points पढ़े हैं in a topic. So first 3 statements are 100% correct. So 4 option हम wrong होगा, right? So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to the channel and stay tuned for the upcoming videos. Thank you.